This video will discuss the single gene mutations, which are part of the fourth key area, which covers all the different types of mutations that can occur. So a mutation is just any change in the DNA sequence that then impacts the protein that should be produced from that gene. So either no protein is made at all, or one that is altered and doesn't function properly is made. So, of the single gene mutations, there are different types. There could be substitutions, where one nucleotide is swapped for another. There could be insertions, where an extra nucleotide is added onto the sequence. Or could be a deletion, where one of the nucleotides is removed. So, the consequences of a substitution mutation, where you've swapped one nucleotide for another, that can result in either a missense or a nonsense mutation. So, a missense, when you've swapped one nucleotide for another, you still get a amino acid added, not necessarily the correct one, but generally has little effect on the end protein. It still is the right length and only one amino acid is affected, but it could still result in a non-functional protein. Alternatively, a nonsense mutation might result from one of these substitutions, and this is where a stop codon is inserted far too early into the sequence, so the protein made is too short to function correctly. Here you can see some examples of this in the diagram. So there is redundancy within the genetic code, and that gives us some protection from these types of mutations. So in the first example, the U nucleotide base is swapped for a C, but because of that inbuilt redundancy, both of these sequences still code for the amino acid leucine. Whereas in our missense, the U has been replaced by a G. Instead of the leucine being added, valine is added in its place. So although only one amino acid has been changed, it can still have a result on the final structure of the protein. Compare that with the mutation. So here the second U base has been swapped for an A, and this is one of those stop codons. If a stop codon is reached, that instructs the tRNAs to stop adding new amino acids to the polypeptide. But because all of the amino acids have not yet been added, protein is way too short to function correctly. The other types of single gene mutations include your frame shift, which result from both insertions and deletions. So frame shifts are when the codons are read incorrectly, so it affects every single amino acid from the point of mutation. So these tend to have much more severe impacts on that final structure of the proteins. So it changes how we interpret, how we read the mRNA molecule, therefore changes all the amino acids that are brought in by the tRNAs, and at the end we've got a protein that just does not function correctly. The last type of mutation we'll talk about here are your splice site mutations. So during splicing, normally introns, non-coding regions are removed, and the exons, the coding regions, are joined together. But in these splice site mutations, sometimes introns are held onto by mistake, whereas exons may not be encoded. So again, this will affect the mature transcript and the amino acids that are added to the final protein, meaning that protein doesn't function as it should do. So let's try now some past paper questions that relate to these single gene mutations. So we're told a genetic disorder of the nervous system results from a mutation in which a nucleotide is inserted into a gene. So which of the following types of mutation causes this genetic disorder? Pause the video and hit play when you're ready to reveal the answer. The correct answer is D. So tells us it's an insertion, and insertions lead to frame shifts. Okay, nonsense and missense, they result from substitutions. Translocations are chromosomal mutations, which will be discussed in a different video. So both insertions and deletions lead to frame shifts. Let's try another. So this time you're asked to explain how substitution mutation may cause the synthesis of a faulty enzyme. Pause the video and write down your response. So the answer to this one, a couple of different ways you could phrase this, but you must make mention of the amino acids. So it's a substitution which tells you one amino acid is changed or a sequence of amino acids is changed because of the mutation. Okay. Remember, an enzyme is just a specialist type of protein. Alternatively, you could say the enzyme is shorter due to the presence of a stop codon. It didn't specify whether it was a missense or nonsense, so either of those options were okay to go with. So either one amino acid was changed or a stop codon was inserted earlier, with the underlying words there being important ones that you needed to include. If you look at the sort of further information as well, so do not accept that active site is changed. So because it mentioned an enzyme, it doesn't need you to talk about your knowledge you have on enzymes. It's merely about the amino acids. This is still about the mutations. So again, hope this has helped with the single gene mutations. Any questions or comments you can always leave below.